The I See Dead Plants podcast shares the stories of people and plants, pests and pathogens, and the conflicts among them. Join us as we speak to the folks who attempt to solve complex problems with plants through pest management research. I See Dead Plants is an extension-based podcast created by the Crop Protection Network and the North Central Integrated Pest Management Center. Music written and recorded by fellow plant pathologist Sean Toporek and hosted by Ed Zaworski.
Welcome back to the I See Dead Plants podcast. As always, I'm your host, Ed Zaworski. Today, I'm joined with by a former classmate of mine, Dr. Will Rutter, and we're going to be talking about root knot nematode, and particularly in vegetable crops, because that's what Will works on. So, how you doing, Will?
I'm doing great. How are you doing, Ed?
I'm fantastic. It's a fine morning here in August. Just a little bit of background information on Will. He did his bachelor's at Knox College in biology and biochemistry, and then went on to get his PhD here at ISU in molecular genetics. And he's currently a research plant pathologist / nematologist with the USDA ARS. All sound right?
Yep, that's all correct. And I guess you probably, I'm down here in Charleston, South Carolina in the Southeast. So.
Oh, yeah. Great. Yeah, that is a good point. It's relevant to... We don't.
Yeah. It's a little bit warmer down here than you guys.
You have root knot nematode too, so.
That's right. Yeah. We have a lot of root knot nematode. That's the big problem down here. So.
I like to start off the podcast just to kind of get to know you, get the audience to get to know you, Will. So, my first question is what is a day in your life like as a nematologist, plant pathologist with the USDA ARS?
Yeah, so I'm here at the U.S. Vegetable Lab in Charleston, South Carolina. And so I work alongside a group of other ARS scientists where we have geneticists who are essentially plant breeders for different vegetable crops. We have pathologists, so we have pathologists working on bacteria, fungi, and viruses here at the station. And we also are adding horticulturalists as well as ag engineers in the near future. So we have a really nice facility. We do run a lot of experiments in both the lab, the greenhouse, and the field to try to understand, in my case, how nematodes interact with and can be managed in vegetable crops.
What are some or all the crops that you work in?
I work on a number of crops. My mandate is to work on nematode problems in vegetables. So as you mentioned, in the Southeast, that primarily means root knot nematodes. And since root knot nematodes have such a wide host range, I work on projects in sweet potato, pepper, watermelon, and cucumber currently.
That's got to be pretty fun.
Yeah, it's cool to be able to work in a lot of different systems. I've learned a lot in the 8 years I've been here. Coming from a field crop background, I didn't even know what a sweet potato looked like, a sweet potato plant looked like till I came down here to the Southeast. And so I've gotten a good crash course in sweet potato planting, harvest, and breeding. So I work a lot with our sweet potato breeder on nematode problems.
Are you a gardener at all? I'm just curious.
I do garden from time to time, but I end up working so much, having so many plants in the field, in the greenhouse, that it's, you get home and the last thing you want to do is plant another pepper plant in the garden. But I do keep a little garden at home for the kids to play in, and harvest stuff, and especially in the springtime is nice. So
yeah, I was just kind of curious. That's working on some of those plants that you do. I was wondering if that would be the case where you're like, I don't want to bring my
it's kind of nice, especially when we're out in the field, oftentimes we'll have leftovers that we're not legally allowed to sell our produce. So, when you have,
sure,
60 bushels of sweet banana peppers like I did a month ago, you're basically giving them away to neighbors and coming up with new and inventive ways to eat them yourself. So yeah, it's
don't really need a garden then, I suppose.
Exactly, right? Yeah.
So, like most people who come on here and I talk to, there's usually a pretty interesting background or an interesting lead up to the realm of plant pathology or entomology or wherever people fall. I guess, how did you become interested in that? Is it something that, you saw at a young age, and not plant pathology in particular, but, I saw you studied biochemistry and biology. Like, yeah, what led you to where you are, I guess?
Yeah, yeah. I mean, like you say, I don't think many kids know what a plant pathologist is, and I certainly didn't. I come from a line of biologists. So my grandfather was a high school biology teacher. My father was a biochemist until he retired a couple of years ago. And so I was always, really into biology and all things biology, whether it was, ecology, playing in in the dirt, looking at, playing with worms out in the field. And then I also my on my mother's side, my grandfather is a farmer down in
Hmm.
southeast Kansas. So I always spent my summers down there, and when I got a little older, I ended up helping him out a few summers. So that gave me a little bit of connection to farming and got a little exposure there. But I did my undergraduate at Knox College, which is a little smaller liberal arts college. So I got some exposure to molecular biology and genetics, and that really got me interested, and I really was interested in genetics and molecular biology. And when I was ready for school, I knew I wanted to go into grad school for genetics or molecular biology. So I applied to a bunch of institutions, and one of those places was Iowa State. So I came to an interview, sat down with a few professors and talked to them about my interests, and they talked to me about the research. And Iowa State was really the only agricultural school I hadn't even thought about before then. But I remember finishing up the interviews that day and being like really excited
Hmm.
about what was going on at Iowa State. And so I ended up coming there in the fall. Man, it was a while ago. Fall of 2008, I think. Yeah. Dating myself now, but then we got a chance to do rotations, and one of the professors who was looking for rotational students was this crazy German dude, Thomas Baum, came in and talked about nematodes and their effectors and how they manipulate plants, and that was sounded really exciting. And so I spent 6 weeks in his lab, and he's the one I stuck with, and it's kind of history from there. And then afterwards, I finished up grad school, and then I moved into another plant pathology lab for a postdoc there at K-State. So I did a postdoc in Edward Akunov's lab on wheat stem rust, so another
Hmm.
biotrophic pathogen, looking at effectors and resistance proteins in that system. And then, ARS was looking for a plant nematologist, and they chose me. So
Awesome.
all history from there. Yeah.
Awesome. What you mentioned your grandfather did some farming on your mom's side in Kansas?
Yeah, yeah. He's still running a cow-calf operation, but he was to when I was a kid, he was still doing row crops, corn,
Corn,
soybean,
soybean?
and
Okay.
wheat. Yeah. So,
Gotcha.
I got some exposure to that.
Gotcha. And then you ended up back in Kansas.
Exactly. Yeah, yeah. And then that dragged myself over to the East Coast. Yeah, yeah. So,
Where did you grow up?
so I actually my family grew up north of Chicago on the Wisconsin border
Oh,
there, Antioch, Illinois. So, I went
not
there through high school. Yeah.
not too far from me in Joliet.
Oh yeah, yeah. My grandparent, my father's parents are actually from Joliet area. That's funny. I didn't realize that.
I think I think you have a really cool and interesting background, like not just straight into plant pathology, but with the interest in like biochemistry and genetics and stuff. I don't know. It seems like that kind of diverse interest set that you have kind of led to. I mean, you'd say better than me, but I think it kind of led to where you are and kind of what you're working on here. It seems like the
Yeah,
perfect
yeah. It
storm.
was it was cool. Yeah, I hadn't. I mean, I knew what a nematode was, but I didn't realize until I got into Thomas's lab how the different aspects of plant parasitism, or how they interact, how intricate the interaction is between a plant parasite and its host. And so that really excited me, and it was kind of an opportunity to look at two different kingdoms of life interacting with each other. We have, instead of just one genome, now you're talking about two genomes and how those genomes interact, on the molecular level, and that really excited me. And then also, I really do like the aspect of, helping farmers. Of course, in ARS, that's really a big portion of my job is to identify ways that I can help stakeholders, in that case, is like farmers and breeders. And so a lot of my job is thinking about how I can translate my knowledge and interest in plant microbe interactions into something useful for our stakeholders that can benefit them. So
Yeah. And I don't know, to me, there's not a lot of better pathogens to do just that than root knot nematode. I
no,
mean,
I agree completely. Yeah.
it's not something that we think about a lot up here, obviously, but man, as far as pathogens go and nematodes go, root knot is a pretty important one, especially in the southeastern United States. So
Yeah. We have a bunch. It's worth noting, when you talk about root knot nematode, we're not just talking about one species either. We're talking, I think last I checked, there were close to, and there's probably more than 100 species that have been characterized now. And so they infect all kinds of different hosts around the world. And, we have 4 or 5 species there in the southeast that are really causing problems, but there's also temperate nematodes too. So like up in the Pacific Northwest, they have Maloidine chipwoody, which is a big problem on potato and other vegetable crops. And then also up north, they have the northern root knot nematodes. That's Maloidine hapla that causes a lot of problems on different vegetable crops. So, we can talk if that's something I think your listeners are interested in, talk about, what the different species are and how they differ and how they're similar, which is, a big aspect of what my research is. So I guess here in the Southeast, historically, we've had the southern root knot nematode, which is Meloidia incognita. And that's probably the most widely spread species. That's all around the world in tropical and subtropical regions. It's the one that's been around forever. But there's also 2 closely related species. So that's Meloidia arenaria. That's usually called the peanut root knot nematode.
Okay.
And then the Javanese root knot nematode. That's Meloidia javanica. And those 3 species, it's kind of, I don't want to get too nerded out on the genetics of it, but are often called the Meloidine incognita group.
Okay.
Because they are so closely related. They're all polyploids. So that means they have multiple copies of their genome. And they all share a single mother genome. So they have one copy of their genome that seems to be almost identical. And then they have a different subgenome that seemed to have come from as yet unknown species. So they have similar effector repertoires, but they tend to infect different hosts and do better in different environments.
Gotcha. So that was my wheels were spinning there. I was just thinking, like, do they infect the same hosts? But so they have like different, if we get back into the Z's triangle, like they have different optimums.
Host range. Yeah. They have they have different, like you can imagine the peanut root knot nematode, and it's known for infecting peanut. But all 3 of these species have massive host ranges. So incognita, the southern root knot nematode, one of the reasons it's so widespread, it's been thought to infect over 3,000 plant species.
Holy smokes.
Yeah, so really immense host range. And then arenaria and javanica aren't far behind, but javanica, there's certain hosts it does a little better against. It's also a little more thermophilic. So it's it does a little better at higher temperatures and reproduces a little faster.
Want to keep up with the latest integrated pest management research? Check out the North Central IPM Center's Pests in Progress webinar. Listen real time or check out the expanding library of past recorded webinars and keep up to speed.
So, Will, for somebody who's listening who's not familiar with root knot nematode, do you want to give like here's what it does to the plant, here's how it lives its life, kind of
Yeah,
just
I guess I'm
basic?
putting the cart before the
No,
horse here. I'm
no,
trying
you're
to get down
fine.
to the molecular side.
This is my job, is to come back and get the clarification stuff. So
Yeah, so root knot nematodes, they're nematodes. They're small, microscopic roundworms. They hatch from eggs within infected soil, and as were referred to as stage 2 juveniles. And those juveniles will migrate to a host plant root, usually following volatile signals secreted by the host plant. And once they get to that host plant, they'll find a root tip, excuse me. They'll basically bury themselves and migrate into the center vascular cylinder of the root.
mm-hmm.
And right in there, they'll choose a few cells in which to secrete proteinaceous cocktail. So they have this sclerotinized mouth spear called a stylet that acts kind of like a syringe, and they can inject individual plant cells with this mixture of hundreds of proteins. And once those proteins are inside that cell, they're going to modify normal host cellular processes and create these what are called giant cells. So these cells grow to several hundred times their normal size. They go through multiple rounds of nuclear division without cellular division. And so you end up with these giant cells with multiple nuclei in them that make them really metabolically active. And that's really favorable for the nematode, because when we say metabolically active, it's acting as a nutrient sink. So it's drawing nutrients in from the rest of the plant and basically acting to feed the nematode itself. And so the nematode will basically produces 7 to 9 of these giant cells around itself, and it will take turns going around feeding off each giant cell individually, and basically remain sedentary for the remainder of its life. And if it becomes a female, it'll turn in this bulbous, almost looks like a giant tick inside the root, and then it'll start secreting a gelatinous matrix and start laying its eggs. So you can have upwards of 600 eggs produced by a single female. And its life cycle, depending on, is very temperature dependent, but most of these tropical species will go through multiple life cycles throughout a growing season. So you get multiple reinfection cycles within the field every growing season, and that can really put a huge parasite load on the plant. In addition to the feeding site drawing nutrients away from the plant, there's also this added effect in the surrounding cells. The nematode induces a tumorous tissue called the gall that can really get quite large. And this can also interfere with normal root architecture. And if you're talking about, something like a sweet potato or a carrot, a root vegetable, that can really it can be severe enough that it'll render it unmarketable. So it can be a severe problem in a lot of different crops.
Yeah, I'll see if I can convince Will to send me some photos that I can share on social media later, but some of them are
sure
very gnarly.
That, yeah. We got some good ones. Yeah. I'll definitely send you some. Yeah.
It's if you've never seen it, the first time you see it, it's a pretty shocking one. There's a lot of nematodes out in the world that they're just doing their thing, and you never know they're there, other than the plant gets stunted or produces less of whatever you're trying to yield off of it. But this one leaves its mark.
Yeah. You got some pretty gnarly roots. Yeah. On sweet potatoes, we talk about seeing pimples on the root, and even though we'll find them in the grocery store often, like just heavily pimpled roots with just and there'll be females inside each of those galls.
I was talking to, I mentioned to Will, I was interviewing Kathy Lawrence from Auburn yesterday. We were talking about reniform nematode. And I hear about agriculture in the South, and it's always just it's like an alien world to me, like how different it is. But one of the things that you mentioned that I was thinking about, as you said, how many generations it can go through. I mean, she was talking about there's times there's places in the South, right, that it doesn't even freeze, and you can have these nematodes basically reproducing all year long. And for something like that has 3,000 different hosts.
Yeah,
I'm
yeah.
sure that it's just like it's nonstop. It's crazy.
Yeah, it's almost impossible to rotate out of it. So it really becomes a numbers game, trying to knock it back with, showing it different hosts, less favorable hosts to reduce the numbers. Using a integrated approach with nematicides is another approach. What I work on, my primary push in my lab is actually to identify new sources of host resistance that I can then provide to U.S. stakeholders to help them try to reduce those numbers in the field.
So this is a perfect transition. I mean, you mentioned that people can sometimes try to rotate, like, or use a trap crop, and there's nematicides. Like, what is the typical management program as of right now for? I mean, yeah, I guess it's kind of a hard question since there are several root knot nematodes you could talk about, but maybe like the incognita group, as you mentioned, is probably the most common or impactful.
Yeah, yeah.
What are
It
people
really depends.
doing?
Yeah, I would say at this point it probably is more dependent on as much dependent on the crop as it is the nematode species you have.
Sure.
But I would say, certainly rotation, picking the proper rotation is important, and not usually a non-host rotation. And there's been a lot of good work by our collaborator down there, Johan Desiger,
Okay.
looking at different cover crops and their ability to suppress nematode numbers from different species across. So a good one that's always talked about is sorghum. Also, cover crops like sun hemp. There are several options that can help a grower reduce those numbers, either between their fall and spring crops or as a substitute. For instance, depending on the species you have, peanut. If you don't have the peanut root knot nematode, Mulligan arenaria, peanut is a good option for reducing. When we talk about the guava root knot nematode, Mulligan enterobiae. So
As many hosts as these things have, it's crazy. Like, I guess if somebody's got a really bad root knot problem, or perhaps let's say they don't know they've got a really bad root knot problem, but they suspect it. How does one sample for these things? Like, are they going out and pulling roots and sending it to a lab? Are they sampling the soil? Like,
yeah, I think both are a good option. So, and most sampling is most effective in the fall, so the late growing season when you still have roots in the field.
mm-hmm.
That's when root knot nematode populations are going to be at their high, and most plant parasitic nematodes will be at their highest. So
Mm-hmm.
that's the best time to sample. But going out, usually refer to your specific state's extension website. Most states, especially in the Southeast, will have a nematode diagnostic clinic, and they have specific requirements for how much soil they need.
Mm-hmm.
But in general, you're going to move across a field in a zigzag pattern and take core samples, ideally with a 1-inch soil probe, and you'll mix those all together, 10 to 20 samples across your field, ideally from within the root zone of your crop plant, and then you mix that together, put it into a sample bag or even a Ziploc bag, and then send it into your nematode diagnostic. And once it goes to that diagnostic clinic, they'll extract those nematodes from the soil, and they'll have trained technicians counting out the different species that are present in your soil. They'll return you a report and usually give you some recommendations about different nematode species, but there's different action thresholds depending on the different species of nematode you have in your field. But usually the action threshold for root knot nematode is one nematode. So if you see one root knot nematode in the Southeast, you should probably take some sort of action
Interesting.
to prevent. Yeah. So
so I'm just curious again. I'm gonna nerd out a little bit now. We see a lot of soybean cyst nematode up here, but it's very different animal, right? As far as like the threshold for sure, but also just there's one species like what's the common practice for like identifying the different nematodes? I mean, I'm sure every lab is a little bit different, but I'm remembering back to my my days of I went and did a workshop on the potato cyst nematode years and years ago, and they were talking about identifying the different cyst nematodes, and you were looking at man, I'm blanking on the name of the structure. You probably know it. On the posterior
the mouth
end.
parts, or yeah, they're using the posterior or the anterior end. So
Yeah,
usually
yeah.
either the mouth parts, obviously, it has to have a stylet and the
Mm-hmm.
shape of the stylet. And then, mind you, I am a terrible morphological
Oh,
diagnostician.
gotcha.
But I
I'm
can
putting
tell
you
you
on
in
the spot.
general, they're looking at, so if you can get a male nematode, they're going to look at the bursa. If
Mm-hmm.
you're looking at female, you're looking at the vulva,
Mm-hmm.
and then for root knot nematodes, and I think cyst nematodes too, they do perianal patterns, which is kind of weird, but they take a really small scalpel and
Yes.
slice the end off the female nematode and put it on a slide. And based on the species, have very different patterns around their anterior end. They're called perianal patterns. And if you look at if you Google it online, you can see some really weird. It looks like an old man, different old men faces.
Yes.
But if you look at enough of them, oh, that one's incognito, that one's arenaria. I'm not that good, so
Oh, I was just trying to remember that perianal because I just remember they were describing them as it looks like a face,
yeah, it does, yeah. Weird little faces, yeah. But
So is your
for
do you
for the rubes like me who aren't very good morphological nematologists, I rely heavily, especially with root knot nematodes. I and a lot of labs rely heavily on molecular tests. So based on their genetics is what I really go by, and then
gotcha?
I'll send it up to a better diagnostician to confirm morphologically if I want to go that far. Yeah.
Gotcha. That was more what I was curious about. Is what is the average lab then is doing it molecularly
Yeah, I
with
would say there's
DNA?
probably a handful of labs in the country I would trust to distinguish between root knot nematode. It's a handful of individuals, and
Hmm.
not even labs, the individuals in those labs who I would trust to make the call between species of root knot nematode in the southeast.
Gotcha.
So most of them are relying on specifically mitochondrial genome sequences. Is that
Gotcha.
as I said, a lot of these root knot nematodes share a common subgenome. So then because of that, there's not enough resolution from a lot of the genome, the actual nuclear genome. So we look at specifically the mitochondrial genome, to distinguish between these different species.
Are you interested in reading the latest integrated pest management updates, such as current pest concerns, management tips, research updates, available IPM resources, upcoming events, and job announcements? Make sure you subscribe to the monthly newsletter, The Central Issue, published by the North Central IPM Center.
You mentioned like having a basically a damage threshold of 1 if you have root knot nematode. If you're a grower of peanuts, let's say, and you send a sample off to a lab and they find a root knot nematode, but they don't know the species. This is a weird question, so feel free to just veto. But if they don't know the species, are they still just saying, okay, we've got root knot nematode, or at that point are they saying let's get more specific and figure out exactly which one this is?
Yeah, it really depends on and certain labs will charge you extra to go down to the species level
Sure.
because it takes that molecular test. But if you're a grower of peanut, and it really depends on and I'm not a great diagnostician. I'm more of a molecular guy, but I would say, you probably have to look at the history of the field.
Sure.
Were you in peanut last year and do you have a high number of nematodes? Well, probably doesn't matter if it's a peanut root knot or nematode or not.
Yeah.
Something's
Okay.
infecting your peanuts, and especially if peanut growers have a very low damage threshold in general because they're talking about a root crop, so, anything and affects the pods, which root knot certainly can. And I know it can also exacerbate some other fungal pathogens in peanut as well, I believe. So, there's a low action threshold for a peanut grower in particular.
Gotcha.
Yeah.
That makes sense. Kind of context clues, field history, what's been going on. There's probably not a lot of people who are like, one day I'm going to grow peanuts. And anyway, just starting at the inception of, and I better see if I have nematodes.
But I mean, any crop you're going, I mean, I'm of the opinion, if you're a commercial grower, it's always good to know your numbers, as the CIS community now is talking about.
Yes.
Actually get, even if you might not think you're having a problem, it might be worth sending it to a lab just to get those diagnostics done and know what you have, what could be causing a problem in the future. Yeah.
So I'm just kind of curious, before I start asking you about your annual review and that kind of stuff, and that was fun to read, but is root knot nematode, like in my mind, hearing like, it's got a bunch of hosts, there's a few different species that have a bunch of hosts, like is this a nematode that's just widespread and everywhere, like it's in every county, or is it localized? Like, is there a preferred soil type, I guess, maybe that you really find it in and that kind of thing? Or
yeah, there is. It's not as finicky about soils as something like reniform seems to be. Reniform nematode has a specific soil type. It seems, or sting nematode seems to have a very specific soil type. Root knot nematode, I won't say it doesn't do better in certain, especially sandy soils. It tends to, just like a sandy loam soil, to do really well in. But it tends to, if it's in a good environment where it has the correct temperature, year-round temperatures, a good host and a set of hosts, and good soil moisture, it's probably going to take off, at some point. And oftentimes, if you're not planting root crops and you're not looking for it, it can go unnoticed. If you're not looking at your roots or you're not checking, it is kind of a silent yield drag on a lot of different crops. So it's not always going to just kill your plant outright. Really, the nematode itself is a biotrophic pathogen. It means it wants to keep that plant host alive as long as possible.
Right.
And so it'll reduce yield until that plant can't take it anymore, or another stressor, drought comes along, and all of a sudden, plants collapse.
Yep. Just like soybean cyst up here, it's the
Exactly,
silent thief in the night, which almost makes it just a little scarier. You can't can't even, you don't even know what's happening.
Yeah.
Well, I obviously I'll put this in the show notes and I'll point people to it. But Will wrote a great annual review article on the root knot nematode, kind of outlining things that we've learned over the years. So I mean, in doing so, Will, I mean, what have we learned about root knot nematode in recent years, or in the entirety of studying root knot nematode, whatever you want to attack?
Yeah. I mean, again, I come from kind of the molecular background, but
Sure.
we've for a long time, I think since the 70s, we've known that, root knot nematodes and other and cyst nematodes were secreting this proteinaceous cocktail into their plant hosts, and it was somehow causing the symptoms and the feeding sites that we could see. But in the past, I would say 20 years, just in the past 20 years, we've started to be able to kind of dissect what those individual proteins are. And there's hundreds of them that we term effectors, that go in and actually manipulate the host plant to cause that damage and enable root knot nematode parasitism. With the advent of genomic sequencing, we started to get a snapshot what these genes are and the proteins they produce, and then kind of one by one different labs have tried to look at specific proteins and how they work in the plant and how they might be advantageous to the nematode. And then with this new generation of long read sequencing techniques, that's even pushed things a little farther. We've gotten better and better genomes. As I mentioned, these are polyploid creatures with multiple copies of each gene, so that makes it very difficult for us to really assemble a really complete genome. But now we're getting these really long read sequencing techniques, and that's helping us out. So now we're getting more and more accuracy and seeing more and more details of what's in these creatures' genomes. And that's giving us hints about how they're able to do what they're doing. Because there's a lot of outstanding questions about how these nematodes are able to infect so many different plant species effectively, cause basically the exact same feeding site to be formed in so many families of different plants. So they're obviously doing something. We've always known they're doing something at a very basal level, interfering with very basic plant molecular processes, but we don't exactly know how. So in a lot of ways that I think about, we could be kind of jealous of them because they're kind of they're kind of done what humans have been trying to do for thousands of years, and that's like create a renewable feed source, manipulate the plants to make a renewable feed source, and they've done so very successfully. So I think there's a lot that remains to be learned about how they do what they do, and things that could be learned. I think could be learned both for mitigating the damage they cause and for understanding how plants themselves function, the basal processes within a plant. So that's what really excites me about looking at an interaction like this.
That is really cool stuff. And I don't know, I've known about root knot nematode since I started studying plant pathology and nematology, but there's things that I didn't realize, like how complicated the genomes of these things are. And it's funny that you said, it's almost hard not to be impressed or like you said, maybe a little jealous of these nematodes. My classes started up this week, and I'm helping teach a forest pest class, and with Laura Isles and Chelsea Harbach in the diagnostic clinic here now. And Laura was talking about the emerald ash borer and like as an example of like, you had like this couple dozen, maybe dozen individuals that came over on some shipping materials and then just like landed in the mother lode of, there's a bunch of defenseless ash that don't have any resistance. And just like these 12 individuals hit the jackpot, and it's hard not to be like, that's wow, that's the definition of success of any organism. Right? and thinking about root knot in that sense is like, man, they've got something going on, and it's exciting. Like, like you said, with the long read sequencing technology, like what can you learn? Like, you just like lay awake at night and just think about the possibilities?
Yeah. From time to time. Yeah, I've lost some sleep on it. Yeah. I won't deny it.
Thanks for listening to part 1 of my discussion with Dr. Will Rutter of the USDA ARS on root knot nematode and the problems they can cause on vegetable crops. Make sure you tune in next week to catch part 2. Thanks for listening, folks.
I See Dead Plants bridges the gap between plant science research and the impact it has on our daily lives. It is brought to you by the Crop Protection Network and the North Central Integrated Pest Management Center, which are both partially supported by the USDA National Institute of Food and Agriculture. For more information on today's topic or other pest management issues, check out our show notes, cropprotectionnetwork.org, or ncipmc.org. Remember, this information is for entertainment purposes only. The statements made in this podcast should be interpreted within the limited context of the particular topics being discussed. Contact your state extension program for local information on pest management. For the full non-discrimination statement or for accommodation inquiries, go to cropprotectionnetwork.org/about. Thank you for listening to I See Dead Plants.